2007 to 13, Associate Professor, Chemical Physics Division, the same university. Again, he has so many honors and awards. In 2019, she got the Jagiellonian laurel for outstanding scientific achievements in field of natural science. 2018, James Rector's Award for the highest score in the students' assessment. In 2018 and 2009 to 18, continuously top high position in the professor's ranking at the same university. 2014, Individual Award for the Ministry of Science and Higher Education for outstanding science achievements. 2005 and 9, Award for the Ministry of Natural Education Science for scientific achievements. 2004 to 8, continuously JU's Rector Awards. Then 2005, awards for the Federal Institute of Breeding, Research, Cultivation of Plants for scientific achievements, Germany. Now, CS membership, a lot of bodies. There's since 2018, membership of Scientific Council of the Institute of Organic Chemistry. Since 2016, member of the Scientific Board of JCET, Poland. Since 2016, member of the Senate Committee for Awards and Prizes, JU, Poland. Since 2016, members of Faculty of Committee Professors Promotion, JU, Poland. Since 2015, Polish coordinator of EU COST, Raman for Clinics, coordinator of STSM. So again, 2008 to 10, scientific secretary of the Faculty of Science, University, same university. Then since 2009 and 10, member of interdisciplinary team to organize JCET, appointed by JU's rector. Then there are so many membership scientific societies. Since 2017, director of International Society for Clinical Spectroscopy, UK. Since 2015, member of the International Society for Plant Spectroscopy, Sweden. Since 2012, member of the Koblenz Society, UK. So he has organized many conferences, international conferences and seminars. He was the chair of coming 2021 International Conference on Advanced Spectroscopy, Poland, coming August 2021, August 22nd to 27. Those who are interested can submit their abstracts for this conference. So nice place, Poland, 2021. August 22nd to 27. So Professor Baranska is the chair of the conference. So Professor Baranska is not new to Kerala. She was here in 2014 at the conference was conducted at the Marijuana College in the International Conference on Perspectives on Vibrational Spectroscopy at Muscat Hotel. He was the keynote speaker on that conference also. He was the member that all committees of different conferences. So with this, I conclude. There are so many other bodies are there. So I just conclude. I now once again welcome Professor Baranska. Now I just hand over this to Professor Baranska. Just you can start, Prof.
Thank you. Thank you, Dr. Joe, for this kind introduction. It's my great pleasure to be with you today and to have a possibility to share with you some of my results with an introduction, rather general one, to Raman spectroscopy. Before I start this official part of my lecture, I would like to mention that I have been 3 times in India. You mentioned one time or one of my visits, but it was 3. 2 times I was in Kerala, at the University of Kerala. It was the time when the conference was organized, and 2 times I was in the city of Bangalore. because also the conferences were organized there. So I have very, very good memories from India, and I'm looking to visit India again in the future. I hope that it will be possible. So let me start with my lecture. At the beginning, I prepared for you slides about my research, and later I added some slides about fundamentals of Raman spectroscopy and Raman imaging. So I hope it will be not too difficult for some of you and not too trivial for some others. So I mixed this information, rather the basic one and the advanced one. So let me start with introduction where I'm working, where I'm coming from. So as you can see, I'm affiliated with Faculty of Chemistry. I hope you can see this pointer I'm using. I hope so. I'm affiliated with Faculty of Chemistry and Jagiellonian Center for Experimental Therapeutics. Both are located at Jagiellonian University in Krakow. I'm also, as you mentioned already, director of the society ClearSpec. At the end, I will present you what is ClearSpec, and I will invite you to the ClearSpec. And I'm also editor-in-chief of Spectrochemica Acta for last few years. So you are also welcome to submit papers to Spectrochemica Acta. We receive quite a lot of papers from India, and I must say that they are really very high quality, so you are very, very welcome. So for some of you not quite familiar, where is Krakow and Poland? So I prepared just one slide, so I'm somewhere located somewhere here. And here is Krakow, the city at the south part of Poland. This is the second largest city in Poland. The number one is our capital, Warsaw. So short introduction, so you can see what you can visit in Krakow. This is the main square of Krakow and the castle, the famous one. Quite close to Krakow we have a salt mine, Wieliczka. Krakow was located many, many years ago on the UNESCO World Heritage List, and some other attractions which are close to Krakow are also located on the list. So when you come to Krakow, you can visit really a lot interesting and historical sites. Quite close to Krakow, we have Tatra Mountains. They are beautiful in summer, in winter, in spring, and in autumn. So anytime it's worth to visit this part of Poland, Krakow especially. Oh yeah, this is what you can do in winter. So I am affiliated with Jagiellonian University. This is very old university, founded in 1364, the oldest university in Poland, the second one in Central Europe, one of the oldest universities in the world. So you can see the main building of the university and new campus, Life Science Park, where I'm located now. So you can see now the Faculty of Chemistry, the new one, where I'm working now. As I mentioned, I'm affiliated also with the JCET, Jagiellonian Center for Experimental Therapeutics. So this is the center I co-developed together with the director of the center for the last 10 years. So since 2010, as you can see. So this is the center where several different laboratories are located, and one of them is Raman Spectroscopy Lab. So this is very well equipped center. We have our own animal house, very broad national, international cooperation, and the main research topic is pharmacology of endothelium. So later on, I will present you the results which were obtained in the center. I will introduce you a bit what is endothelium and why we are interested in this pharmacology of endothelium. But as you can see, we have a multidisciplinary approach, so Raman spectroscopy is just one among others. We have access to other techniques and to other labs in one place. So the lecture agenda for today is a few words about Raman spectroscopy past, today, tomorrow. I will not talk much about tomorrow because I realized that it would be enough to say what we are doing today with some hints for tomorrow. And then I will present you results which were obtained by my group in the relation to diagnostic, detection, and pharmacology. All of them somehow related to endothelium, as I mentioned, and that give you the impression what kind of study we can perform using Raman spectroscopy, what kind of information we can obtain. So I must say that Raman spectroscopy, and probably most of you know that, is really very universal technique. So we are using the technique for studying pharmacology of endothelium. But if someone else is interested in any other topic, I'm sure that Raman spectroscopy can be used. So it's really very, very universal. So Raman spectroscopy belongs to the group of spectroscopy techniques. So techniques which investigate interaction between sample and light. And what we are measuring by Raman spectroscopy is light scattered by the sample. So generally in spectroscopy we can use a broad range of light, and we can investigate broad range of interaction, and one of them is light scattering. So this is, I could say that if we would use another detector, another detection technique, at the same time we would be able to measure spectra of other spectroscopies of other technique. Probably you can see here also infrared spectroscopy. So this is a complementary technique to Raman spectroscopy. I will not talk today about infrared spectroscopy. However, this is also the technique used in our group. And here we are measuring the light which is transmitted or absorbed by the sample. So what can we detect, how we can see the sample using Raman spectroscopy? I need to stress that I will talk today only about the vibrational Raman spectroscopy, not about a rotational one. So if we are talking about Raman vibrational spectroscopy, it means that we look at the molecule, and specifically we are looking at the motion of this molecule. So as you can see, molecule makes various vibrations. We know the number of these vibrations, and this vibration can be described by laws, by rules, which we know from the physics. So these movements are somehow similar and can be described in a similar way. And as a result of the measurement, Raman measurement, we obtained Raman spectrum, vibrational Raman spectrum, which provide us information about all this motion of the molecule. So as you can see, this simple molecule is described by this somehow complicated spectrum, and each band from this spectrum provides us information about the type of the motion of the molecule. And based on this spectrum, we can recognize that we have that molecule in our system. We can also obtain information about the quantity, about the interaction of this molecule with the environment, etc. But the most important thing I would say is identification. So based on these bands, we know that the molecule performs specific kind of vibration, and based on that we can identify it. So when we look at the theory, at the energy states, in the context of Raman spectroscopy, we are talking about Stokes Raman scattering and anti-Stokes Raman scattering. However, what we are measuring generally in Raman spectroscopy is this Stokes Raman scattering. So this is inelastic process in which vibrational energy is changed. And you can see also infrared absorption, as mentioned before, as a complementary technique, which also provides information about the changes which occur between vibrational energy states. Today we meet because of Raman, who is presented here on that slide. But this is not only Raman himself presented here, but this is also the first Raman spectrometer, as you know, probably even better than me. So this is how he discovered the effect of inelastic scattering, how he measured the effect. And this is not only the first Raman, this is the first Raman spectrometer, but Raman himself plays very important role here, because he's a kind of detector in this spectrometer. So with that system, he was able to observe inelastic scattering of light by sample, which was put here. And he obtained a Nobel Prize for that. I am showing you two of my spectrometers which we are using nowadays, almost 100 years later, a bit less. So FT Raman spectrometer and dispersive spectrometer with many different lasers and different microscopes equipped. So this is only part of our lab. The first, or not really the first, but almost, but one of the first Raman spectrum taken by Sir Raman, you can see here on the top. And at the bottom, you can see how the spectrum can be obtained using modern Raman setup. So with FT Raman, this is the example how the spectrum would look when you perform the measurement with FT Raman. And you can of course decrease amount of the sample, decrease accumulation time, and improve resolution, what is extremely important. So this band can show you that it is much complicated, much complex. So you can also obtain information about isotopes. So when we are talking about Raman spectroscopy, then we need to remember that behind there are various Raman techniques. And this is a short list, really a short list of various Raman techniques. All of them are based on Raman effect on inelastic scattering of light. All of them are used by us in our lab. We have such equipment, or we are performing the measurement in collaboration. So when I will not talk much about these techniques, just to mention. So if we have resonance Raman spectroscopy, then we can enhance effect, because I didn't mention, but most of you will probably know that inelastic Raman scattering is very weak effect. However, when we have in the sample a chromophore responsible for absorption of the light, then we can perform the measurement, which is called resonance Raman spectroscopy. So we are going to the first electronic excited state, and the light which is scattered is much higher intensity in comparison to the normal Raman scattering. The first Raman map, because we are talking today a bit about history, so let's look at the first Raman map. I'm interested in Raman mapping, Raman imaging, so this is why I'm showing you this slide. So this first Raman map showing the distribution of some specific compounds, chromate and cyanide distribution in this complex sample was obtained in 1975 and published that year in Journal of Raman Spectroscopy. This is very important journal for Raman spectroscopists. However, today we have many other journals, as you know. So by using a specific bound, we can look and we can obtain the distribution of specific compounds. So this is the simplest approach how to obtain Raman map or Raman image. Here is the idea behind Raman mapping or imaging. Before we used rather mapping word, but nowadays more and more we are talking about Raman imaging. So from the sample of interest, from the area of interest, we perform the measurement point by point in the XY direction. From each point, what is very important, we obtain the full spectral information from each point from the sample. We have a full spectrum which can be analyzed. And the basic approach is that we select a band of interest, for example, that one, and then we obtain a chemical map showing us the distribution of, for example, that band, which is associated, which is correlated with some specific compound. If we select another band and perform the integration of that band, then we obtain another chemical map showing us information about another compound. So here is the example how it does not work. Sometimes it works, sometimes not. Oh, sorry. Okay. So no movie, but let's move to confocal Raman microscopy. Nowadays, I must say that all Raman microscope or spectrometers have the possibility of confocal measurement. What does it mean? It means that we can focus on specific level of the sample, and we can collect information from this specific layer. So we can measure the top of our sample, or we can focus in the on the mid layer, or the information can be obtained from the bottom. So by using the approach, we can really obtain, we can penetrate sample relatively deeply and obtain information not only from the surface. So let's move now to biological application of Raman imaging. So before I will show you some results, I would like to put your Raman imaging system among other Raman imaging systems used for biomedical purposes. So here, this, as you can see, these various imaging systems were divided according to the resolution of the method. So for some methods, we can obtain rather more general information. I mean, the information at lower resolution, but there are some methods, and Raman and fluorescence imaging belongs to these methods, where we are able to obtain information from cellular or molecular even level. So from this slide, you can see that with Raman imaging, we are able to obtain information about individual organelles from the cell, or even sometimes some specific molecules. To study human body, generally we have 3 electromagnetic windows where the light is attenuated. From one side, this is X-ray. From the other side, it is MRI, and somewhere between we have optical methods. And Raman spectroscopy belongs to this mid group of methods. So by using one or the second or the third group of methods, we can obtain different kind of information from about human body. What is quite important, this is to remember how the depth of penetration is correlated with the resolution of the method. So we have several methods already used for medical, biomedical purposes. And this method, as you can see, can relatively deep penetrate our sample, but the resolution of this method is not so high. Where is Raman spectroscopy? It's somewhere here. So you can see that this is the method which provides us similar type of information in a sense of penetration or resolution as just mentioned, confocal microscopy to photon microscopy, stimulated emission microscopy. So you can see that with that, Raman spectroscopy is, I would say, really dedicated for a single cell analysis, can be, of course, used also for tissue analysis. So this is the paper which we published a few years ago. We collected data of Raman spectroscopy measurements obtained from cross-section from various organs, and we compared this data with a single spectra of tissue homogenates. So with that paper, we wanted to show that, first of all, Raman spectroscopy can be used to study various organs successfully, that we can find really specific spectra profile, which give us a lot of information about these various organs. And we also wanted to say that even if we cannot perform cross-section, or we don't want to perform cross-section analysis because it's time-consuming, also single spectra from tissue homogenates are different for these organs, so also can be used to differentiate organs. However, of course, cross-section analysis with Raman imaging provide us detailed information about these organs. So let's move to endothelium. I will go rather fast through next slide because I see that yes, time is going very fast. So in the center where I'm working, we focus on endothelium. So endothelium is a single layer of cells covering each artery. This is kind of interface between blood and tissue. Single layer, this is quite important. So on one side, we would say that, what is single cell? It's almost nothing. But when we when we realize that it covers each artery, then the endothelium, then we need to realize that endothelium covers a large surface up to a few thousand of square meter. It's small, single cell is rather small, about less than 1 micrometer thickness. But in total, our body contains about 1 kilogram of endothelium. But we are focused on endothelium not because it's so heavy or that it covers so big surface, but because of the unique function in our organism and about organ-specific mechanisms. So depending on the organ, the function, the composition of endothelium is different, and we want to learn as much as possible about that. What is really motivation of our research? This is first of all the correlation between endothelial dysfunction and development of various diseases. Number one are cardiovascular diseases. So here you can see the normal situation and normal endothelium, and here you can see the pathological state with the dysfunctional endothelium and many, many other microscopic changes occurring. We are talking about primary endothelial dysfunction, but we need to remember that there is also secondary endothelial dysfunction associated with various diseases. So it can be a cause of various diseases, but it can be also a result of various diseases. So we are working on various models in vivo, ex vivo, in vitro study. I will show you some results. And the last point I would like to mention that there is a correlation between biochemistry of endothelium and some alteration in blood cells, blood particles. So for the diagnostic purpose, I would say that it's not necessary to look at the endothelium, but to look, for example, at blood cells. However, to treat various diseases, we postulate, not only we, that endothelium can be the aim of the pharmacology. So through endothelium, we can treat various diseases. Yes, so one more example to show you that if we are really able, if we would be able to detect endothelial dysfunction, so this is really the marker which would provide us information about the development of atherosclerosis at a very early stage. So as you can see, here is the development of atherosclerosis, and the usual procedure is EKG, which is performed when, which provide us information about the development of the diseases, but at a late stage. So some other techniques are also dedicated for analysis, but at relatively later, more advanced stage in comparison to the situation when the first symptoms of the diseases can be detected in endothelium. So let's move to our lab. As mentioned, we are working on animals. We take various organs, take cross-section, and perform the measurement from the cross-section, mapping, Raman imaging, infrared imaging. We take tissue homogenates, blood plasma, we work on cell line cultures, and quite important, for the last few years, we developed new protocols of isolated primary cells. So these are cells which are taken from various organs, from the tissue. I will show you some results and explain why this approach is so, in our opinion, important and should be developed. So let's move to a few examples which were taken by my group members. First few slides about development of atherosclerotic plaque and some other diseases like diabetes, hypertension, cancer. So here you have a movie showing you how this, how such atherosclerotic plaque is formed. You can see this plaque here. So this is the process where first the lipids are accumulated, various lipids, and at some point we see also calcification and then clot. So we took a cross section from the aorta. So this is the aorta. We took a part of the aorta, a small part of the aorta for imaging, parallelly to other methods. So this is the small parts of aorta cross section imaged with Raman spectroscopy. And here, yeah, this is the results which were published in Biophotonic journal. So you can see how Raman spectroscopy see atherosclerotic plaque. So this is one example. The other example, so here you see calcification very nicely. So all what you see here are Raman images analyzed by special tools. I will not talk today about methods of analysis. This would be a separate lecture. How do we analyze Raman spectra? Because not always the analysis, or I would say that very seldom the analysis is easy and based on the marker bands. Sometimes we need to take the full spectrum for the analysis and look for very small differences between the spectra to extract the difference between various components. So another example where you see the calcification of aorta and the spectrum taken from the spot. So the spectrum is really specific. You can see that we can recognize the cholesterol from cholesterol esters also in the atherosclerotic plaque. And this is the example where we investigated as unstable atherosclerotic plaque. So this is the situation very dangerous for our life because as long as the plaque is stable, is fixed to the aorta, then it's not so bad situation. However, when this fibrous cap is very thin, then the atherosclerotic cap can be released and can block in the aorta. So with Raman spectroscopy, we can measure what is the thickness of this fibrous cap, how stable or unstable is this plaque. And we can use Raman spectroscopy and here additionally AFM approach, so we can measure really the thickness of this fibrous cap. So you can see what is the what are the dimensions of this fibrous cap. So here you can see that we are able to measure the same part of the sample with various methods. Oh, sorry, yeah. So here is the staining and Raman maps showing us the distribution of cell nuclei, elastin fibers, and this too correlates with histochemical staining. I told you that endothelium is a single layer of cells. So here is the aorta, and here with Raman spectroscopy we were able to identify this single layer of cells. So this red part. From this red part, we can extract spectra from diabetes model and control, and you can see that spectra are really different. That the composition of endothelium from diabetes model show us significant differences in comparison to the control, and the same we have shown for hypertension and for cancer. So we can really, from single spectrum, from single cell of endothelium, we can really say if this is the control endothelium or the control, or if it comes from the disease. So another example, this is the study we are started a few years ago, and this is quite important part of research we are doing now. We are trying to understand the mechanism of drug-induced toxicity in endothelial cells. So maybe I will show you the comparison. So endothelium covers each artery as a single layer, and these cells are relatively small. And as I showed just before, the approach we are using to measure endothelium, it is not so easy because of this small size and also some other limitations that we need to use animal models. But we can perform measurements using cell culture in vitro analysis. So then we have cells of much bigger size. So cells, endothelial cells, which are cultured, they can have the size approximately 20-30 micrometers, so they are really huge. And what is the advantage? We do not need animals, and we can perform various model studies. So we can study the development of the disease, and we can also study the influence of drug on endothelium. So this is a part of our study. So you can see that we investigate the mechanism of action, different type of action of drug to cells. So the first one, quite known one, oxidative stress. The other one, phospholipidosis, endoplasmatic reticulum stress, DNA damage. So there are several compounds or drugs which we know from the literature that they have a specific mechanism of action. We know that. And we ask ourselves if we can find spectral markers of these various mechanisms to detect this activity of these drugs on the cell. I mentioned here about cytocardiotoxicity because this influence of these drugs on the cell usually is somehow toxic. The level of this toxicity we can measure, so we do the study parallelly, of course, with other biochemical measurements and tests. So if we are measuring single cells, we need to use confocal microscopy. So one single cell can be measured. This is our setup. Can be measured from the bottom to the top, so layer by layer. So this is how it looks, more or less, layer by layer. And then we can select the band of interest, and to present the distribution of this compound of interest. So let's go very fast through these results where we study various mechanisms in cells. So oxidative stress, number one, this is the mechanism then where we have imbalance between antioxidants and ROS. So this is the process which is goes together with inflammation, apoptosis, tissue injury, very complex. However, when we look a little closer to the complexity of this process, we can find some chemical changes. I mark these changes in red, which in fact are able to be detected by Raman spectroscopy. So changes or damage even of protein, DNA, and some changes related to cytochrome. So here you can see how complex is the experiment. So we take a cell, we treat the cell with a specific compound, menadion. This is the compound which we expect to produce oxidative stress in cells. Cells are put on calcium fluoride slide. They are cultured on that side. We measure live cells and fixed cells. We measure cells with different lasers. So there is really a lot of variation. With, of course, we perform the measurement with different incubation time, with different concentration of menadion. And then, here you can see different techniques which are used parallelly to Raman spectroscopy. And at the end, we perform the analysis. So at the end, we see a single cell and we see organelles of these cells, chemical information about these organelles, and how these organelles change upon the treatment of menadione or another compound. So here you see that I mentioned before that we need to be careful to select the proper time of incubation and the proper concentration, not to trigger apoptosis or necrosis in cell. Or if we want to analyze such processes, then we can of course use such conditions, as you can see. So anyway, this is the spectra. You can see that single spectra taken from cells from some specific from each point of the cell are very good quality. From such spectra, we can really have rich information about the composition and changes of composition. So from the analysis, here you can see the condition of the experimental condition. We concluded that we can correlate with oxidative stress, decrease of the content of cytochrome, of nucleic acids, and phenylalanine. And by changing the laser, we move from the resonance effect of cytochrome. So we were able to see more information about lipids. We were able to have a nice lipid feature. And we could additionally say that with oxidative stress, we observe increase of some lipid bands and decrease of some proteins. So, so here is the comparison of these two measurements. It's kind of summary as I just mentioned. So from this PCA analysis, from these two loadings, we can obtain the same information as just mentioned by me. So I will not go in detail about the biochemistry of these various processes which we investigate on cells caused by various molecules or drugs. I will show you the spectra to see what kind of information we obtain. So this is about endoplasmatic reticulum stress, where we were mainly focused on proteins and changes due to proteins. So for first we used tunicamycin, the drug known to cause this endoplasmatic reticulum stress. In this case, we observe some changes. However, tunicamycin, even in very low concentration, accumulated in really high amounts in the cell, so it was a bit difficult to obtain information about other changes, but still we obtain we were able from the spectra and from the analysis to obtain some spectral markers for endoplasmatic reticulum stress, so the change of phospholipids and proteins. For another drug, tapsigardin, we have noticed exactly the same spectral changes as before, so we could correlate these changes with endoplasmatic reticulum stress. Phospholipidosis, another example. So this is a group of drugs, cationic amphiphilic drugs of different action, of different application, where we expect to observe the changes related to phospholipid, accumulation of phospholipids in cells, increased synthesis of phospholipids. And indeed, yeah, I will not talk about the mechanism, but so here you can see a few drugs which were used for this study to look for spectral markers of phospholipidosis. Again, very complex experiment. And we were able to notice in the spectra increase of the intensity of the bands related to various phospholipids. So indeed, in this case, the marker of phospholipidosis is can be found in Raman spectra. Oh, sorry. And the last example, DNA damage. So we did quite a lot with anthracycline antibiotics, like doxorubicin and daunorubicin. These two compounds were used for the measurements in very low concentrations, as you can see, nanomolar concentrations. And even such low concentration of the drug used to incubate with cells was detected by us in the Raman spectra. So when we used dead excitation, we have observed increase in the background of Raman spectra. And based on this increased background, we were able to localize daunorubicin, doxorubicin in the nucleus, detect this compound at very low concentrations. We found that daunorubicin is even more toxic because at lower concentration can be detected in the nucleus of the cell. So we perform very detailed analysis to look precisely at the distribution of daunorubicin, doxorubicin in the nucleus. This is the control where no drug was used and no, of course, was detected. So we calculated the ratio occupied by daunorubicin or doxorubicin in the nucleus, and from these results, from this ratio, we confirmed that daunorubicin is more toxic, so accumulates in lower concentration and more than doxorubicin. We obtain also some information about the changes of DNA. So there are some specific bonds found in the spectrum. So we found that strong accumulation of doxorubicin is related with increase of a DNA marker of doxorubicin DNA interaction. By changing the laser, we were able to see some other changes in the cell due to doxorubicin or daunorubicin. So these changes were described here. So the last part is about primary cells. I mentioned you that this is quite new approach. I mean, for the last few years, but still quite new. So I will present you the example about the study on non-alcoholic fatty liver disease, but the measurements were performed on primary cells. So what are primary cells? So first of all, cell cultures, as mentioned before, are very valuable complement to in vivo study. But to understand the signals that governs intracellular communication between cells, primary cells are a better biologically relevant model. So what it means? So instead of working on cell line cultures, we try to isolate cells from organs and to put them to the culture. Sometimes it is possible, sometimes not. But anyway, we try to analyze cells which were isolated from specific organs. So this is the procedure. Just a few slides. So it will not. This slide will not provide you all steps, but as you can see, we take the organ, and the organ is homogenized. And then through many steps which were not presented here, we obtain a kind of soup of cells, and from this soup of cells we separate specific cells, like here for liver cells, for example, hepatocytes, from liver endothelial cells, and some others. And then the cells are put for the measurement, Raman imaging, and for the analysis. So we are working on primary cells isolated from the liver, like endothelial cells and hepatocytes. We work on primary cells isolated from heart and from blood, eosinophils. At the same time, we are still working on cell lines, and we see a lot of differences between these two. So this is also the reason that if only we can measure on primary cells, then we select primary cells for our study. So here you see non-alcoholic fatty liver disease problem. So this is a normal condition of our liver, and this is the non-alcoholic fatty liver with a lot of lipids accumulated. So these red spots were stained with all staining. And with Raman spectroscopy, we trying to learn as much as possible about the biochemical changes, but not only about lipids, of course, also, but also more specific changes also occurring in endothelial cells. So somewhere here we should localize endothelial cells. Yeah, so the study which we perform on this model was performed on endothelial cells and on hepatocytes, which were isolated from the liver. And we perform the study on the progress of this disease. So it was not just one measurement, but a few experiments where we follow the disease, the progression of non-alcoholic fatty liver disease. So here you can see kind of soup, as I mentioned before, of here a soup of cells. And you can see that the spectra of specific cells are different, especially endothelial cells can be differentiated from the other cells. Hepatic stellate cells are very characteristic. They contain vitamin A. Hepatocytes are quite rich in lipids, and some specific bands can be also identified for endothelial cells. So from that, not going into details, I just wanted to show you how the analysis is performed. So for endothelial cells and for hepatocytes, from many cells, measure also using depth profiling. We extract chemical information, which is summarized here, to follow the changes of these chemicals during the development of the disease. So I will not go in detail because maybe it's not so important for you what kind of changes we observe, especially in the disease, but just to give you the impression how the images of single cells look like and what kind of information we can obtain. So we concentrate mainly on DNA, RNA, on lipids, of course, and we quantitatively try to follow the changes upon the development of the disease. So this is the second week of the disease, the 15th week and 20th week of the disease. This is quantitative analysis for the second, 15th and 20th week for endothelial cells and hepatocytes, and some conclusions which I will not read now because I see that I'm talking really too long. So to summarize, I would like to say that our group and other groups are doing quite a lot in the area of biomedical application. And this is mainly kind of basic research. But from this basic research on some specific cells, some molecules, at the end we hope to have a few clinical study or application even. So it is done a lot here, but we are looking mainly what will drop here in the future. So with that, I would like to finish, to invite you to International Society for Clinical Spectroscopy, non-profit organization. You can find it here. You are very welcome to join. This is the organization to gather researchers, students, to discuss, to promote the translation of spectroscopy into the clinical environment. So you are welcome here very much. And as mentioned at the beginning, we are organizing next year a conference in Krakow. You are also very welcome to come. I hope that it will be possible for all of us to meet in Krakow, but if the situation will not improve enough, then the meeting will be organized virtually. So anyway, you are very invited to join us at that conference, if not in real, then at least virtually. So some attractions close to Krakow. And the last slide of acknowledgement. This is part of my group. This is quite old photo, and some people from the JSET we collaborate, and some funding which allow us to perform the research I present you today. So with that, I would like to thank you and to switch.
Yeah. So it's a time for asking questions. Participants can raise a question. If you want, you can ask the question. Any question, please? You just put in a chat box or you can ask.
If not now, maybe you can write me. Yeah.
Yeah.
I see the questions about PhD. Yeah.
Yeah. Yes.
So I must say that during the last few years and even now, we have open positions for PhD. So at the moment, I have a few persons from abroad who joined me. So I have 2 PhD students from abroad. So I had also a postdoc. So I hope that we will have more open position. So please contact me. If only I have open position, then I will share with you this information. Oh, Professor Rastogi, I can see you.
Yeah. Professor Rastogi is there.
Very nice.
Yes. So any question, please? Is there any other question? Okay.
Yeah, actually, hello, Professor Baranska. It was a very nice presentation. Yeah, so I think this field is quite different, but what I understood, I'm Mamta from physics department. I'm a theoretician. I'm working on non-equilibrium statistical mechanics and biophysics related stuffs. So here you just mentioned about the defective the DNA cells that the liver cells that the spectroscopic I mean that means can you just briefly explain more about it little bit, that what exactly, how it helps the.
I'm afraid I did not hear the second part of your questions. So I have heard you presented yourself, but then I didn't hear you really.
Yeah. Mamta, please post your question in the chat box. Dr. Mamta?
Yes.
Please,
Yeah.
please.
So what I mean, is it like that defective DNA damage response? I mean, how the Raman that spectrum, how it helps, and is it like the DNA damage response kind, and then repair means in that direction? Is it like that?
I heard a little only. So I heard that you asked about DNA damage. So
Okay.
I don't hear the second part. So with Raman spectroscopy, I must say that as you know, as you have seen, we can really. I cannot say maybe easily, but we can detect DNA band, which can be assigned to A DNA, B DNA, and some changes if occur in the system related to DNA, like interaction with DNA, can be observed. So what I showed you, this was looking for the marker of interaction between compound and DNA, and that can be done. So the band is not very intense, of course. So this is not so easy. The other point is that in fact we are looking not only at DNA but also RNA. So to compare these two, then we can obtain better information about the oxidative stress in cells. The other point which I didn't mention during the lecture is also the quite new approach which we are applying now, which we are developing now, is introducing to cells some molecular reporters which go specific to some organelles. So, for example, to nucleus, and then we can obtain information about the changes due to the DNA in a non-direct way, but with better sensitivity. So I probably this is not the full answer to your question because I didn't hear you, but yeah, if you have more questions, please contact me by email.
Yes.
Maybe I can explain you better.
Sure, I think that I got
about the
answer.
preparation of cells.
Yeah,
because now I can read. So I see the question about the preparation of cells. So this is really nothing really complicated what we need to prepare. So cells are cultured on the calcium fluoride substrate, and for some measurements we are using live cells, but then the measurement cannot be longer than a few hours in total. So if we want to measure high number of cells, or we want to perform some additional experiments, so then it's better to measure fixed cells. So usually we perform both live, and then we work on fixed when we see that there is no change upon the fixation.
Any more questions? Okay. So I would like to add one more thing. Professor Baranska is the chief editor of Spectrochemic Acta. So during, I think Professor Baranska is the chief editor-in-chief. So those who are interested, Spectrochemic Acta is one of the leading journals in spectroscopy. So it's a very good journal, high impact factor, nearly 2.7 or 8 something now. So it's a good, highly impact factor journal, good research journal. So those who are interested, you can send papers to this journal. Now, just Professor Subodh will give the Professor vote of thanks.
Yeah, you are very welcome to submit papers to Spectrochemica Acta. We got quite a lot from India, and as I mentioned at the beginning, they are really high quality.
Yes.
What I would like to stress that sometimes before you submit your paper, please have a look at the scope of the journal, because Spectrochemica Acta is focused on spectroscopic methods, optical spectroscopic methods, and the development of these methods. So you really need to show, yes, what is the development of these methods. So I'm saying that because it's a pity when people submit paper and they are presenting only NMR data or EPR data. They forgot that this is not the scope of Spectrochemica. So you can include such data, but still optical spectroscopy methods are the main scope.
Okay.
Yeah.
Thank you, ma'am. Thank you. Okay. So, Professor Subodh, unmute your. Okay.
Yes, I'm here. Thank you, Professor Baranska, for your wonderful talk on Raman imaging, and we have listened to a different topic. It's a truly interdisciplinary one, and hope all our participants got many information from your talk. Thank you so much, and I would like to extend my sincere thanks to all the participants, and most importantly, the talk was organized by Professor Hubert Joe and our head of the department, Dr. CBKS. I would like to extend my thanks to them also. Thank you, Professor Baranska, once again, and hope to see you in Krakow next year. Thank you.
Yeah, you are welcome to come here to visit us, please.
Okay. Thank you so much, Professor Baranska. So, Professor Rastogi, you want to talk to me? Professor Rastogi? Professor Rastogi? Hello? Hello, Professor Rastogi?
Yeah, I can see you, Professor.
Please unmute your mic, sir. Professor? Yeah, okay. Professor? Okay. Okay. Thank you very much, ma'am, once again, for giving spend a valuable time for us.
So sorry for taking you so much time, and sorry for this delay.
No problem. No problem. Yeah.
It's a new experience for me with this meet, but yeah, next time will be better.
Okay, thank you. Okay, thank you so much for my invitation to give a very useful talk for us. Okay, thank you so much.
Thank you.
Thank you. Okay.
Okay, bye bye.
Okay, thank you. Bye. Thank you. Okay, bye.